This video explains primer extension capture, or PEC, a technique that allows the targeted retrieval of DNA sequences from extremely low copy number, highly fragmented DNA sources. We have used this technique to sequence complete mitochondrial genomes from Neanderthal bones over 40,000 years old, and the method can be used for capture of any small target regions or single nucleotide polymorphisms and from many individuals. The main steps in this procedure are PCR amplification of a primary DNA library, quantification of the amplified library by quantitative PCR, target primer extension on the amplified library, bead capture of primer extension products to remove non-target fragments, and elution and sequencing of the captured and washed targets.
Hello, I'm Adrian Briggs from Svante Pääbo's lab in the Department of Genetics at the Max Planck Institute for Evolutionary Anthropology in Leipzig. Today we're going to show you a procedure for isolating specific DNA sequences of interest from a complex, highly degraded DNA source, such as ancient bones. We have used this procedure to study the mitochondrial genetic diversity of Homo neanderthalensis, or Neanderthal man. Let's get started.
The DNA used in our experiment comes from Neanderthal bones between 40,000 and 70,000 years old, which were found at 4 sites across the Neanderthal geographic range. A bone is first exposed overnight to UV light to decontaminate the surface of any extraneous DNA. A fine drill is used to produce between 100 and 500 milligrams of bone powder. DNA is extracted from the bone powder using a procedure described in the 2007 article Comparison and Optimization of Ancient DNA Extraction by Nadine Rochland and Michael Hofreiter. The DNA extract is then converted to a sequencing library by the addition of universal adapter priming sites to the end of all DNA fragments. Because a 454 library generated from a Neanderthal bone contains only tiny amounts of DNA, sometimes just a few picograms, this primary DNA library is first amplified by PCR, using emulsion PCR primers corresponding to the universal library primers. To prepare the PCR mix for library amplification, combine the following reagents in a PCR tube. Use 10 microliters of 10x GeneAmp PCR buffer 2, 10 microliters of magnesium chloride, 2 microliters of BSA, 1 microliter of DNTP mix, 3 microliters each of 454M PCR forward and reverse primers, 1 microliter of AmpliTac Gold DNA polymerase, 25 microliters of library template, and finally add 45 microliters of PCR grade water to yield a total volume of 100 microliters. AmpliTac Gold DNA polymerase is stable at room temperature, so it's not necessary to work on ice. After preparing the PCR reaction, place the PCR tube in a thermocycler, starting with a denaturation step of 95 degrees Celsius for 12 minutes, followed by 14 cycles of 95 degrees Celsius for 30 seconds, 60 degrees Celsius for 1 minute, and 72 degrees Celsius for 1 minute. At the end of these 14 cycles, set a 72 degrees Celsius 5-minute final step, after which the PCR tube can be set indefinitely on 10 degrees Celsius until ready to use. After PCR amplification is complete, follow it with purification of the PCR product using a Qiagen Minolute spin column, according to manufacturer's instructions. Elute DNA from the column with 50 microliters of EB buffer supplied with the Minolute kit. Before primer extension and product capture, quantitative PCR should be performed on aliquots of the unamplified and amplified libraries in order to quantify and check the quality of amplified library product. The 14-cycle amplified library should contain several thousand times more DNA than the primary library. If it does not, amplification conditions should be checked and repeated. Primer extension capture primers, which will be used to capture the sequences of interest from the amplified library, should be designed like normal PCR primers to match a desired target region with an ideal melting temperature of 60 degrees Celsius. To the 5' end of each of these target sequences, add this 12-base tail sequence and include a biotin residue at the 5' end. Up to 150 PEC primers can be used together in a single multiplex mix, made from 200 micromolar stocks. For the primer extension reaction, prepare a PCR mix in a suitable PCR tube by adding 5 microliters of 10x GNAMP PCR buffer 2, 5 microliters of magnesium chloride, 1 microliter of BSA, 0.5 microliters of DNTP mix, 5 microliters of the primer extension capture primer mix, 0.5 microliters of TAC Gold DNA polymerase, 2 microliters of the 14-cycle amplified library DNA, and finally 31 microliters of PCR-grade water to yield a total volume of 50 microliters. If the amplified library template contains more than 1 × 10⁻12 copies per microliter, the template should first be diluted with EB buffer so that not more than 2 × 10⁻12 copies are added to the reaction mix. After preparing the PCR tube, place it in the thermocycler and set it for a single reaction run, starting with a 12-minute denaturation at 95 degrees Celsius, followed by 60 degrees Celsius for 1 minute, 5 minutes at 72 degrees Celsius for primer extension, and when the single cycle is finished, 72 degrees Celsius to hold the PCR product until the next step. In order to avoid non-specific primer annealing and capture, directly pipette 150 microliters of PBI or PB buffer from Qiagen Minolute purification kit into the PCR reaction tubes, while the reaction mixture remains in the thermocycler held at 72 degrees Celsius. Doing this inactivates the polymerase while the temperature is too high for non-specific extension of the target primers. Close the tubes, vortex briefly, and then spin the liquid down with a benchtop centrifuge. Use only PBI or PB buffer in this step, as other binding buffers like Qiagen PM buffer can interfere with the downstream capture process. Next, remove the mix from the PCR tube and transfer to a Qiagen Minolute spin column for purification according to manufacturer's instructions, as previously done. Elute DNA from the column with 30 microliters of EB buffer supplied with the Minolute kit. In order to remove all fragments that have not annealed to PEC primers, a bead-based capture step is performed. First, resuspend a stock solution of M270 streptavidin beads by vortexing and remove an amount of beads equivalent to 25 microliters per sample. Wash the beads twice with 500 microliters of 2 × BW buffer and resuspend in the same volume of 2 × BW as the volume of beads originally taken. Pipette out 25 microliters of this suspension for each sample. Add 25 microliters of the purified eluted DNA to 25 microliters of the bead suspension, saving the remaining 5 microliters for later quantification. Mix the DNA and bead suspension by pipetting up and down and stirring with the pipette tip, and then rotate slowly on a benchtop rotator for 15 minutes at room temperature. After mixing, transfer the contents to a fresh 1.5 ml PCR tube. During the previous rotation incubation step, some non-target DNA may have become stuck to the tube wall, so transferring the bead suspension to a new tube avoids the possibility of this non-target DNA being released into the final products during the heat elution step. Using the magnetic particle collector, pellet the beads. After 10 to 20 seconds, all of the beads will have migrated to the wall, leaving a clear supernatant that should be removed. To wash the beads, add 500 microliters of 1 times BW buffer to the tube and vortex for about 3 seconds at slow speed, once with the tube upright and again with the tube inverted. No beads should remain sticking to the tube wall. Using a tabletop centrifuge, spin down the suspension for 1 to 3 seconds, just long enough to draw all liquid from the walls of the tube. Return the tube to the MPC and wait 10 to 20 seconds until all the beads are collected at the side of the tube. Then remove the supernatant. The first wash is now complete. Repeat this procedure a further 4 times. Following the last 1 times BW wash and supernatant removal, add 500 microliters of 1 times hot wash buffer. Mix the suspension by briefly vortexing and spinning down as before. And then shake the tube for 2 minutes at 65 degrees Celsius on a thermal block shaker at 1000 RPM. Following a brief spin, remove all traces of the supernatant within 1 minute of removal from the thermal block. This step will remove fragments that may be binding to PEC primers but were not extended on in the primer extension step. Resuspend the pellet in 30 microliters of EB buffer and transfer the mix to a fresh 1.5 ml PCR tube. Incubate the tube in the thermal cycler at 95 degrees Celsius for 3 minutes. Pellet the beads in the magnetic particle collector. It should take 10 to 20 seconds for the supernatant to clear completely, and remove the supernatant to a fresh labelled tube, taking care not to transfer any beads. Keep the supernatant, which contains the captured product of DNA fragments. The beads can be discarded at this stage. To amplify the captured product, prepare a PCR mix to a total volume of 100 microliters, as done in part 2, and perform the same PCR cycling conditions again. After PCR amplification of the captured product is complete, follow it with purification using a QIAGEN Minolute spin column as previously done in section 2. Elute DNA from the column with 50 microliters of EB buffer. The product is now ready for high throughput sequencing. We designed 574 PEC primers to tile across the whole Neanderthal mitochondrial genome. We split these into 4 multiplex mixes and performed 2 serial rounds of primer extension capture on libraries from 5 Neanderthal fossils from 4 sites across Eurasia that are dated between 40,000 and 70,000 years old. Generating between 170,000 and 500,000 sequences per individual and aligning the results to a mitochondrial reference sequence, we found that the PEC procedure had massively enriched for mitochondrial DNA, achieving complete coverage across the mitochondrial DNA for each individual at an average of between 18 and 56.3-fold coverage. With these results, we could generate a high-quality complete mitochondrial genome sequence for each individual. We then performed a phylogenetic comparison of these sequences with a worldwide sample of present-day humans and our closest living relatives, the chimpanzee and bonobo. This confirmed previous findings that Neanderthal mitochondrial DNA is closely related but clearly distinct from that of modern humans. Furthermore, comparison between the Neanderthal sequences themselves revealed extremely low genetic diversity in Neanderthals, lower than among humans worldwide today and similar to present-day Europeans. This evidence, combined with an analysis of protein evolution in the Neanderthal sequences, suggests that Neanderthals had a long-term low population size, which is consistent with archaeological evidence suggesting that Neanderthal population density never reached the levels achieved by modern humans.
We've just shown you how to perform primer extension capture to amplify and capture target sequences in preparation for high throughput sequencing. When following this procedure, it's important to follow all the temperature specifications on the thermocycler, but be aware that primers can have different annealing temperatures, so you may have to adjust the annealing temperature to suit your particular primers. And take care during all the washing steps to remove as much liquid as possible. So thank you for watching and good luck with your experiments.